Hi, my name is Katie Cheetah, and I'm a postdoctoral fellow in the labs of Dr. Rich Mooney and Dr. Fan Wong at Duke University.
And I'm Valerie Michael, and I'm a graduate student in Rich Mooney's lab.
And today we're going to tell you about recent work we did in which we identified a specialized population of neurons in the midbrain that are essential for the production of social vocalizations in the mouse.
But first, a bit of background.
Since the dawn of time, animals have used vocalizations to communicate.
Okay, we exaggerate, but it is true that innate vocalizations are used ubiquitously throughout the animal kingdom for things like defending territory, attracting a mate, and so on. We humans also use innate vocalizations, in particular to signal our emotional states, both the good and the bad.
Given the importance of vocalizations to animal communication, our long-term goal is to understand the neural circuits that support the production of vocalizations, and herein lies the challenge. It has been known for decades that a structure in the midbrain known as the periaqueductal gray, or the PAG, plays an important role in the production of vocalizations.
However, the PAG is a functionally heterogeneous structure that's also involved in pain processing, defensive behaviors, and autonomic regulation. Thus, it remained unclear whether or not the PAG contained a specific population of neurons dedicated to controlling vocalizations, and if so, how this population of neurons contributes to vocalization.
To overcome these challenges, we turn to the humble and genetically tractable lab mouse. Mice emit a rich repertoire of ultrasonic vocalizations, or USVs, in social situations, especially during courtship. For example, when a male mouse is pursuing a female social partner and its USVs are pitch shifted into the human hearing range, it looks and sounds like this. To identify and selectively manipulate neurons in the PAG of the mouse that are active during the production of USVs, our overarching strategy was to use an intersectional genetic method that was developed by Fan Wong's lab called CANE, or Capturing Activated Neuronal Ensembles. This method relies on a knock-in mouse in which expression of a destabilized TVA receptor is driven by activation of the promoter for the immediate early gene FOS. Hence, the TVA receptor is expressed in a similar spatial and temporal pattern to that of FOS. Second, the CANE method relies on the injection of designer viruses that can selectively infect TVA-expressing neurons, hence enabling the permanent and selective expression of genes in neurons that were recently active during the production of USVs. Using immunohistochemistry, we confirmed that the mouse PAG contains neurons that upregulate FOS in mice that recently produced USVs. We called these neurons PAG-USV neurons, and we next used CANE to bidirectionally manipulate the activity of these neurons to test their functional role in USV production. First, we used CANE to express tetanus toxin in PAG-USV neurons to irreversibly silence them. Prior to viral injection, male mice produced high rates of USVs toward female social partners. Following viral expression, male mice were muted for USV production. Notably, these males still engaged in non-vocal courtship behaviors, consistent with the idea that PAG-USV neuronal activity is selectively important for USV production. Second, we used CANE to express channel rhodopsin in PAG-USV neurons to test the hypothesis that activation of these cells is sufficient to elicit USVs. Indeed, optogenetic activation of PAG-USV neurons elicited USVs from male mice, even in the absence of any social partners or cues. Next, we sought to understand how PAG-USV neurons contribute to vocalization. Just like humans, mice vocalize during the expiratory phase of breathing. Thus, to drive the timing of individual USV syllables, PAG-USV neurons would need to shape the pattern of a mouse's breathing. However, careful measurements revealed that the optogenetic activation of PAG-USV neurons did not change the mouse's ongoing pattern of respiration. In fact, USVs could only be elicited during the post-inspiratory phase of breathing, consistent with a role for PAG-USV neurons in the gating but not the patterning of vocalization. Finally, we characterized the descending projections of PAG-USV neurons by using Kane to label these cells with GFP. We identified a prominent projection to nucleus retroambiguus, a premotor respiratory region in the caudal brainstem. To test the functional role of this projection, we optogenetically activated PAG neurons that project to nucleus retroambiguus. Activation of these neurons elicited USVs in both male and female mice, underscoring the importance of this midbrain to hindbrain projection to vocalization.
In conclusion, we identified a subpopulation of PAG neurons whose activity is both necessary and sufficient for the production of mouse USVs. These neurons gate vocalization, but their activity does not dictate the individual syllable pattern. Finally, we showed that the projection from PAG to the nucleus retroambiguus plays an essential role in vocal gating in both male and female mice.
We suspect that similar neurons reside in the PAG of all vocalizing vertebrates, and that the pathway we've uncovered plays a highly conserved role in vocal communication. Furthermore, now that we have genetic access to the PAG neurons that control vocal communication, we have a tractable entry point to study the brain-wide circuits for vocal communication.
Thanks for listening, and feel free to contact us with any questions.